Now this is the second last video in Key Year 17, looking at something called genetic drift. So when we're going to be looking at genetic drift, you have to think first of all about, say, a total population. And we talked before about when you've got a population, you've got all sorts of different genes that are in there. That's called the gene pool. Now, if you've got a population that's large, then the alleles transmitted during the reproduction process is normally really large as well, and so you get a full representation of all the alleles present in the gene pool being passed on down through the generations. So if there's no environmental change or no unusual factors, the genetic composition of a large population stays fairly constant from one generation to another. And this is what is meant here by this part here, which says if a species is under no selective pressure, frequencies of individual alleles will stay the same from generation to generation. But if that's the case, then you get something called genetic drift, and we're going to look at some of the reasons behind that. So first of all, what do we mean by genetic drift? Well, what we mean by that is that it's a random increase or decrease in the frequency of genetic sequences. And it occurs, well, we're going to look at three different reasons behind that. We're going to look at sampling error, neutral mutations, and founder effects. So sampling error. Now, sampling error, really what we're talking about here is a small population. And small populations are very prone to genetic drift because the samples of alleles transmitted to the next generation is normally relatively small, and so it's often not representative of the gene pool as a whole. Some of the alleles may be overrepresented and others might be underrepresented. So imagine here we've got, on this diagram here, we've got a population and there's a 50-50 number of alleles for the green and for the beige. Now when it comes to the fertilisation process, that is a random process, so it's mere chance. So there's a 50-50 chance every single time fertilisation is taking place of a beige allele being selected or a green allele being selected. Now that doesn't mean that you'll get an even number of green and beige ones. What it means is that sometimes what happens is, in this case here, you'll get more beige ones being drawn for reproduction than the green ones. And so you can think of this in terms of, if you've got a small population, it's maybe a case of, the green sperm fertilises only green eggs, or the beige fertilises only beige eggs, and this time round more beige sperm have fertilised more beige eggs because it's just a matter of chance. So what happens is you get a second generation here where there are more beige alleles in that generation. There's been a drift here towards the beige alleles. And then here the same sort of thing happens. More beige alleles get selected, and so therefore what happens is you get a drift here towards more beige alleles in the population. Now it could be that eventually this sort of scenario happens where more green alleles are selected, and therefore what happens is you then get this drift towards these green alleles over here. But this is what we're talking about when we're talking about genetic drift. And sometimes what might actually happen is, if we say we don't have this scenario here, but this time round what happens is it's just the beige alleles that get selected for, and therefore the fourth generation ends up with only beige alleles 100%, and there's zero green. Then you've actually lost that allele from that next generation, and it's never going to appear again. So you've had this drifting effect that's taken place. Now the second example we want to look at in genetic drift is this thing here called neutral mutations. Now, if you remember from your gene mutations before, if you remember, neutral mutations are where you've got a change to the nucleotide sequence of a gene, but it doesn't change the amino acid that is coded for. So an example of that would be, say you've got a sequence GCC, and there's a neutral mutation that takes place where the C is replaced with an A at the end of there, so you get a GCA. Now both of these code for an amino acid called alanine. So because you've got that neutral mutation there, there's no change to the protein that is made, and so there's no change to the phenotype. So it doesn't affect how well adapted an individual is to the environment. So they say that neutral mutations are not subject to natural selection because there's not any change that's taken place there, so it doesn't really matter. But it says here that they are affected by genetic drift. Well, what we mean by that is, for example, if you've got a small mutation that is passed down through generation after generation again and again. So in the example that we saw previously, say for example, in the population you've got individuals that have got this nucleotide sequence, and then you've got ones that have got this mutated nucleotide sequence. Now, looking at the example that we had before, where we were looking at sampling errors, it might be that more of these genes here end up in the population just due to genetic drift. And so you end up with an increase of these here and a decrease of this here. So you get this subtle change that's taken place in the gene pool over a period of time because of genetic drift. So that is what they mean by neutral mutations being affected by genetic drift. And many geneticists believe that most changes that occur in the frequencies of these neutral genetic sequences are as a result of genetic drift acting as a mechanism of evolution. What that means that over a long period of time you get changes in the genetic sequences that occur. And eventually there could be, say, a loss of this sequence here, and this sequence here starts to end up in the species over a very long period of time. So that's, in this here, we're going to be looking at a founder effect. Now, a founder effect is where the population becomes isolated a section of the population becomes isolated from a larger population, and that population doesn't have the entire gene pool. So you'll have gene frequencies that are different in that population. Now it's easy to look at that in this diagram form here. So you've got a population of organisms here, and you'll see there that there's green alleles, there's red, there's yellow, there's a bluish colour. And what happens with this, if you've got several generations happening and there's no environmental change, then that original population stays fairly relatively stable. But if you get a small group of organisms that break away, a splinter group, and they become isolated from the main population, that may be due to something like some sort of physical separation. Maybe there's a flood and a river or something like that forms, and it separates this group here away from the original population. Now you'll notice here that that splinter group does not have the same alleles in it. It's just got the red and the yellow and the green there. So therefore, that what happens over time, the members of the splinter group, as they reproduce, then what gets produced is a whole new population, which is different from the original population, and there's been this change in the frequencies of gene sequences, and there's actually been the loss of an allele in that population. So we see that after several generations, the genetic makeup of this new population is very different from the original population, and its members tend to become distinctive from the other population or the original population. Now, in this example, we've only really looked at one gene or one allele that's ended up being lost from the population. But in reality, you'll have many alleles of many genes that would probably be involved in this sort of thing. And an example is looking at this example here. It was thought that North America was first populated by a small group of Asian people who migrated across the land bridge, which is now the Bering Strait. And then they became isolated from the rest of their original population. And the founder effect is believed to account for the differences in the percentages of the population possessing certain blood groups. And you'll see this here. So the group here, the original group that is thought that these Native Americans came from were Chinese. And you'll see the percentage of blood groups there for A and B and AB and O group. But when it comes to the Native Americans there, you'll see that there's a much, much lower percentage of blood group A and B, and there's actually no blood group AB there present in the population. And there's this very high percentage of O blood group. So it looks like really what's happened there is that small splinter population that broke away, the majority of them had these alleles here, and over time that has continued to be so. Now, I want to have a look at that alters a population's genetic makeup, and it occurs as a result of the bottleneck effect. Now, this is covered in Unit 3 usually, but I thought it would be useful just to talk about this just now under this heading as well. Now, the bottleneck occurs when something like a disaster, such as a fire or a flood or an earthquake, acts in an unselective way. So this bottle here is representing a population here, and the original population. And what happens is that you have this earthquake and it wipes out a large amount of a number of the population. And if the surviving population is very small, it may have lost much of the genetic variation needed to adapt to future changes that are there. And such a population is said to suffer from this bottleneck effect. So here we've got original population here. Some sort of event happens that results in this bottleneck effect here, where only some of the population survive. So like, as I said, a flood or a fire or something. And so you end up with this surviving population. And you'll see that the container representing the surviving population has got less genetic variation, and the allele frequencies are different from the allele frequencies that were over here at the beginning. So this loss of genetic diversity, what happens is you really produce a population whose members are so similar that reproduction amongst them is genetically equivalent to inbreeding. And since inbreeding results in poorer rates of reproduction, the population may become extinct, or it might slowly recover and survive. And there are some certain species, such as the northern elephant seal, they've got a low genetic diversity among their members, yet they still manage to remain or survive. Another example of this is in cheetahs. So it was thought that 10,000 years ago, something like that, there was a bottleneck effect which wiped out all but a handful of cheetahs. and it's been found that cheetahs are very close genetically. So these close relatives, the ones that were left, they had no option but to mate with one another. And so that inbreeding has led to this loss of genetic diversity over the years. It's been discovered recently, though, actually, in amongst female cheetahs. They're found to be very promiscuous, and they mate with lots of other partners, other males. And it's thought that the benefit of that behaviour is that the female undergoes induced ovulation each time she mates. That is, she releases an egg every time she mates. And therefore, when she produces a cub, each of those cubs in that litter are actually from a different father. And that process helps to ensure some of the genetic diversity in the surviving population. It's also thought it might actually protect the cubs from being killed by an adult male cheetah, because the males that are in that area won't know whether that cub is theirs or not. But that's your bottleneck effect. Now, I will put a video on iTunes U explaining the differences between the bottleneck effect and the founder effect, so you don't get confused by both of them. And we will look at it again in Unit 3.